In the last video, we studied a model in which stochastic messenger RNA transcription and deterministic messenger RNA degradation generated Poisson distributed messenger RNA copy numbers. In this video, we find that messenger RNA copy numbers are also Poisson distributed in a more realistic model in which both transcription and degradation are stochastic. We return to the inside of a cell and describe transcription in the same way as in the first video. A copy of RNA polymerase repeatedly encounters a sequence of DNA, occasionally succeeding in producing messenger RNA, but much more often than not, failing to initiate transcription. Many unproductive collisions typically take place between rare instances of transcription. The collisions are independent. We concisely represent this cartoon using a wheel of fortune. The outcome of spinning the wheel once is a model for the outcome of a bunch of encounters between the copy of RNA polymerase and the gene of interest. If we land in the yellow sector, a single transcription event has occurred. We have described transcription as a stochastic event by considering it as a rare outcome of many repeated molecular encounters. We depict messenger RNA degradation as a stochastic process in the same way. A strand of messenger RNA may collide with an endonuclease multiple times before being degraded. Perhaps the copy of messenger RNA survives one collision because the strand is tangled into a format difficult for an endonuclease to grab. Perhaps another collision fails to initiate degradation because the wrong side of the participating endonuclease collides with the strand of messenger RNA. The number of collisions that must take place, and thus the duration of time that must elapse before a particular strand is degraded, is not necessarily the same as the lifetime, measured from transcription to degradation, of any other strand. Some strands might be longer-lived, while other strands might be degraded sooner. For this model, we assume that collisions between a strand of messenger RNA and the endonuclease are statistically independent. In other words, the number of collisions that a copy of messenger RNA has already survived has no bearing on the likelihood that it survives its next encounter with the endonuclease. In this case, the survival times are exponentially distributed. Now that we have developed mathematical representations of the two mechanisms that constitute our model, we will arrange them on a timeline to understand the statistics of instantaneous copy numbers of messenger RNA. As in the first video, we determine the number of copies of messenger RNA that are counted at a time t count by considering preceding times at which copies of messenger RNA might have been generated. Both this video and the previous describe transcription in the same way. A line of wheels is to be spun, and those spins that land on the yellow sector individually represent a single transcription event. To determine whether copies of messenger RNA are not only transcribed but also counted, we investigate how long messenger RNA molecules survive. For example, suppose that the spin associated with time t source produces a copy of messenger RNA. The exponential distribution we just discussed enumerates a variety of durations that this copy of messenger RNA might survive. If the dye lands on 1, 2, 3, or 4, the copy of messenger RNA is degraded before it can be counted. But if the dye lands instead on 5 or 6, the copy of messenger RNA is present in the cell at time t count. The probability of being counted is associated with the golden height under the exponential curve, and the probability of not being counted is associated with the blue height above the curve. 1/3 of the probability is associated with being counted, and 2/3 with not being counted. If a copy of messenger RNA is generated at time t source, it has a 1 out of 3 chance of surviving long enough to be counted. To determine whether a copy of messenger RNA originating from the time interval centered around time t source will be counted at time t count, spin the wheel at the bottom to determine whether transcription occurs, and then, if it does, spin the wheel at the top to determine whether the copy of messenger RNA produced survives long enough to be counted. We want to sketch the wheels that we would spin if we needed to determine whether copies of messenger RNA generated at other times survived long enough to be counted. Reflect the exponential decay to run from right to left, and then slide the resulting plot toward the right so that its vertical edge sits flush with the dashed line indicating t count. The height of this new curve as it passes through time t source specifies a golden height below and a blue height above, with both of these distances equal to the corresponding double-headed arrows previously drawn at time t count. Again, the relative proportions of these golden and blue heights indicate the relative sizes of the yellow and blue sectors in the wheel at the top. Because this geometric conclusion has not relied on the numerical value of t source, we can say that the ratio of the golden height to the blue height determines the likelihood that messenger RNA survives long enough to be observed at time t count, even as we sweep t source toward earlier times toward the left and even as we sweep t source toward later times toward the right. In the same way that the height under the yellow curve decreases for earlier and earlier times, the wheels we have just generated contain less and less yellow as we move toward the left. The wheels labeled by the yellow brace at the bottom describe the chances of transcription, and the wheels along with the plotted curve, together labeled by the golden brace, describe the chances that molecules of messenger RNA survive long enough to be counted, assuming they are transcribed at all. Taking both transcription and survival into account, what is the contribution of a given time interval to the copy number of messenger RNA molecules finally counted at time t count? For this time interval, the probability of a transcription event is 1 out of 20. In that, on average, 1 out of 20 times that a copy of messenger RNA is generated, the likelihood that it subsequently survives at least long enough to be counted is 1 out of 3. Together, this means that the probability that this time interval generates a copy of messenger RNA to be counted at time t count is 1 out of 60. A third of a 20th is 1 out of 60. Multiplying the probability of transcription and the probability of survival gives the probability of contributing a copy of messenger RNA that will eventually be counted. The newly generated array of wheels at the top of the slide looks different from the kinds of arrays of wheels we might most be familiar with from discussions of Poisson distributions. The wheels we have generated have a variety of sizes of yellow sectors. As we will now explain, we nevertheless obtain a Poisson distribution. The yellow sectors on wheels A and B have roughly the same size. The yellow sector on wheel B is a little smaller than on wheel A, but as a crude approximation, we simulate the process of spinning wheels A and B by spinning, instead, 2 copies of wheel A. The next 2 wheels, C1 and C2, both have noticeably smaller yellow sectors. The chances of obtaining a single messenger RNA count by spinning both of these wheels is roughly equal to the chances of obtaining a messenger RNA count by spinning a copy of wheel A. The next 3 wheels, D1, D2, and D3, individually have even smaller yellow sectors. Spinning these 3 wheels has roughly the same probability of generating a messenger RNA count as does spinning 1 copy of wheel A. The yellow sectors in wheels E1, E2, and so forth are becoming so slender that spinning these next 9 wheels has roughly the same chances of generating a messenger RNA count as does a single spin of a copy of wheel A. We are approximating spinning a lot of wheels with yellow sectors of different sizes by spinning, instead, a smaller collection of copies of wheel A. To see that this is an approximation, look at wheels D1, D2, and D3. These 3 wheels together have about the same likelihood of generating a copy of messenger RNA that will be counted as does the single copy of wheel A above. However, spinning wheels D1, D2, and D3 can, in principle, also generate 2 or 3 copies of messenger RNA that will be counted, whereas spinning the single copy of wheel A can never generate more than 1 copy of messenger RNA to be later counted. We neglect this discrepancy because the likelihood of getting more than one of the D wheels to land on yellow is exceedingly rare. It's difficult enough to get any one of the wheels to land on yellow. Because each of the wheels at the bottom of the slide is meant to represent a bunch of molecular collisions, each can be replaced by a collection of wheels representing a smaller bunch of molecular collisions with a correspondingly smaller chance of generating a transcription event, illustrated using correspondingly smaller yellow sectors. By making the yellow sectors at the bottom of the slide very thin, we can make very small the kind of error we have just said that we make by approximating wheels D1, D2, and D3 with one single copy of wheel A. We said that the height under the yellow curve corresponded to the probability that a copy of messenger RNA could survive long enough to be counted. The almost completely yellow wheel at the right is a cake. It's actually blue at the core, but it has been mostly decorated with yellow frosting, and the source of the frosting is the tube whose height equals the height under the yellow curve. For this other time, the height under the curve is less, the tube of frosting is shorter, and so a smaller fraction of the blue cake is covered in yellow. The amount of frosting in the tubes decreases moving from right to left, so increasing fractions of blue cake surface remain exposed moving from later toward earlier times. The viewer may verify that the area under this exponential curve remains finite even when the left-hand boundary of integration is allowed to move to arbitrarily early times. Even though cakes could be drawn into the arbitrarily distant past, there is only enough yellow frosting to justify sketching a finite number of copies of wheel A. For this example, grouping wheels using pink braces only allows us to justify drawing the 5 copies of wheel A we have already illustrated. There's not enough yellow in wheel F1 and so forth extending beyond the left edge of the slide to justify drawing even another copy of wheel A. What are the statistics that describe spinning a finite number of identical wheels arranged unevenly across a timeline? They are the same as the statistics that describe spinning the same finite number of identical wheels arranged evenly across a more condensed time interval. The statistics remain unchanged when we say that these spins of wheels are a subset of a larger set of spins and that we have neglected the spins outside the pink dashed box. This is the mathematical model we described in the first video. As in the first video, there are rounding errors resulting from coarsely regarding wheels of fortune touching the boundaries as being either completely inside or instead completely outside the pink box. When the wheels are replaced with wheels representing even smaller time intervals, these edge effects are diminished. However, we leave the illustration at its current scale so that the icons of wheels of fortune remain visually clear. In the top half of the slide, we have illustrated a collection of identical wheels associated with uneven durations of time. We obtained this diagram by analyzing a model for messenger RNA dynamics with stochastic transcription and stochastic degradation. In the bottom half of the slide, we have illustrated a collection of identical wheels associated with equal intervals of time. We obtained this diagram by analyzing a model for messenger RNA dynamics with stochastic transcription and deterministic degradation. In both examples, a Poisson distribution characterizes the number of spins that land on yellow sectors for a collection of wheels spread out across time, and the same Poisson distribution characterizes the messenger RNA copy number counted at an instance in time. The assumption that messenger RNA has a lifetime connects the probability distribution of transcription events occurring over time intervals to the distribution of copy numbers observed at individual times. The viewer may wish to use a master equation approach to verify that stochastic transcription and stochastic degradation lead to a Poisson distributed steady-state distribution of messenger RNA copy numbers. Can you check this conclusion numerically using the stochastic simulation algorithm? It is important to remember that the purpose of this slide deck is to practice explaining the distribution of copy numbers of a molecule by thinking about the stochasticity of synthesis events. The purpose of these slides is not to claim that these toy models are correct in every instance in which experimental single-cell molecular copy numbers appear to be Poisson distributed. While inconsistency demonstrates that a model is false, consistency does not demonstrate that a model is true.